So in this chapter, chapter 11, we're going to look at how cells communicate with each other. So this was actually figured out that cells do communicate with each other by looking at bacteria. We realized that there were external signals that were getting converted into responses within the cell. So cells would identify mates through chemical signaling. We would say that a cell that is transduced has its surface changed into a form that brings about the response of mating. So we noticed that bacterial cells would try and mate with cells that other cells interested in mating. It wouldn't go after the ones that were not interested. So your signal transducing pathway is when a signal on the cell's surface gets converted to a specific cellular response through a series of steps. And the studying in bacteria has been useful because it's very similar in yeast, mammals, plants, and bacteria. So what would happen is the bacteria would send out a signal that they were interested in mating, and another bacteria would pick up the signal that said it was interested in mating, and that's what would lead to those cells going about that mating procedure. Bacteria are also quorum sensing. They can sense the concentration of signaling molecules from other bacteria in the area, and this allows them to sense the local density of cells. This can lead to coordination of activities in the population. It is also important for our health. By having large numbers of bacteria in certain areas, this will discourage the growth of pathogens. So one of the reasons why your gut does not always get salmonella every time you consume a single salmonella cell is they are able to sense that there are other bacteria in the area, and it will discourage growth. It also helps with forming biofilms, which are aggregates of bacteria that a lot of times are going to form their own recognizable structures in regions of specialized function. An example of where you would have a biofilm is on your teeth. So signals can be local and long distance. A couple examples of local signals here would be these gap junctions between animal cells or plasmodesmata between the plant cells. They're also useful for cell-to-cell recognition, so the cell can recognize a marker on the outside with a receptor it has. Both animal and plant cells have cell junctions that directly connect with the cytoplasm of adjacent cells. So animal cells can actually communicate by direct contact of surface molecules in cell-to-cell recognition. So some of our regulators are going to be local. They're going to influence cells that are in the local vicinity. So one example is growth factors. These are in animal cells, and these compounds can actually stimulate nearby target cells to grow and divide. With paracrine signaling, you can have numerous cells that will receive or respond to molecules of growth factors produced by just one cell at the same time in animal cells. So this allows one cell to almost be the boss in the situation and tell the other ones when it's time to grow or stop growing. With synaptic signaling, this will happen in animal nerve cells. You have an electric signal that's going to trigger the release of a chemical signal, such as a neurotransmitter, which will pass that message over and stimulate the target cells. So hormones can be used for long-distance signaling in both plants and animals. So in animals, we have the endocrine system, where you're going to have the message travel through the circulatory system. In plants, you can have plant growth regulators. These can travel through vessels or diffuse in the air as gas. So one of the more common gases in plants is going to be ethylene gas. This will trigger fruit to ripen. We can actually use this if we pick fruit and store it that is unripened. If it's picked green from the tree, it can be shipped easier that way. They can be given ethylene gas in a warehouse that will trigger them to ripen and then be taken to the store at that point. When you have transmission of nerve signals, this can be a way of long-distance signaling. So a nerve signal could travel from your head down to your toe. So when we look at stages of signaling, there are 3 stages to it. Reception, where the target is going to detect the signal molecule and binds the receptor. 2 is transduction. This is going to convert the signal to a form that brings about specific cellular responses. And then 3, the response, which actually triggers the specific cellular response. So rather than getting bogged down in too many details in this chapter, I want you to really keep these 3 parts in mind. We're going to look at different ways that you can have reception occur, different ways transduction can occur, and different ways that you can have the response get triggered. So you want to keep big picture in mind and don't worry too much about the details. You want to basically know the different ways this can happen. So when we look at reception, this is when you're going to have the signal molecule bind the receptor protein, and it's going to change its shape. This is going to involve a ligand, which is a molecule that specifically binds to another, and it will often cause the receptor to change shape. So you can have receptors in the plasma membrane. Most of these will bind a specific site on the receptor protein, and then it will transmit the extracellular information into the cell. Couple examples, we see this with G protein kinases and coupled receptors and the tyrosine kinases. The intracellular receptors, these are in the cytoplasm or the nucleus of the target cells. So this means that signal molecule is going to be small enough and hydrophobic to cross the plasma membrane. For example, nitric oxide or steroid hormones can do this, and they can end up controlling the genes. They can act as transcription factors, which are proteins that are going to turn genes on. So G protein coupled receptors, these are going to be in the plasma membrane, and they're a receptor that works with G protein that binds GTP. Some bacteria actually make us sick by interfering with these G protein receptors. So you've got this G protein receptor here, your plasma membrane, this inactive G protein, and an enzyme. When the signal molecule comes in, it's going to activate this receptor. It's going to use GTP, and that is in turn going to activate the enzyme and carry out the cellular response, and then it will be released. So these are going to be used temporarily and get reused. They're going to shut down rapidly so that you can send the signal more than once if it's needed and keep the cell alert. So receptor tyrosine kinases, these are characterized by having enzyme activity. They're going to attach phosphates to the tyrosine. A kinase is just an enzyme that's going to catalyze the transfer of phosphate groups. So anytime you see a kinase, that's going to be part of how the enzyme works. So here you've got the signal molecule and the ligand binding sites that are going to have these tyrosines on there, which is how they get their name. So when the two signal molecules are going to bind to these receptor tyrosine kinase proteins, they will come together and form a dimer. So that activated dimer then is going to transfer and become phosphorylated, and then it will activate the relay proteins that are going to pass the message on inside the cell. Another type of message we can send is using the ion channel receptors. We can have ligand-gated ion channels. So here you've got this channel where the gate is closed. And your signaling molecule is going to be the ligand that's going to come in and open the gate, allow the ions to cross, and when it leaves, the gate will close. We can also have these be voltage-gated ion channels. We use these in the nervous system. So instead of it being a signal molecule that comes in and opens the gate, it would be an electric impulse. So you can see this if you use electric stim on somebody's skeletal muscle. You can artificially put in the electric impulse, and it will open the gated ion channel and send the message for the muscle to contract. So we can also have intracellular examples here. For example, the steroid hormone testosterone. So the testosterone will diffuse into the cell and attach to the receptor protein and make your hormone receptor complex. This will come in and attach onto the DNA and affect transcription, making the mRNA, which will lead to a new protein being produced. So next we're going to look at transduction. These are the cascade of interactions. This is going to be how we pass that signal into the cell using a series of relays. Most of these are going to have multiple steps, which will allow for greater amounts of amplification of the signal and also more opportunities to have coordination and regulation occur. So we can have signal transduction pathways. These are going to bind a signal molecule in the plasma membrane, And the receptor will trigger a chain of molecular interactions that leads to the response. Most of the time, that original signal is just a trigger. It's not actually going to enter the cell. So with protein phosphorylation and dephosphorylation, you're going to use protein kinases and protein phosphatases. So the protein kinase is going to transfer phosphate groups from ATP to the protein. Most of them are going to act on proteins different from themselves, unlike the tyrosine kinases. So many of these relay molecules in the signal transduction pathway are protein kinases that will act on other protein kinases in a phosphorylation cascade. So a lot of these will regulate cell reproduction, and abnormal activity of these can lead to cancer. So the protein phosphatases, these are going to be the enzymes that are going to remove the phosphate groups from the protein. It will dephosphorylate them. So it provides a mechanism for turning off the signal and makes that protein kinase available for reuse. So what happens is you're going to have these protein kinases where you're basically going to pass that phosphate group from one molecule to the next along a chain, which is your phosphorylation cascade, until you get to the active protein. Second, messengers. These are going to involve small molecules and ions. So you'll have a small non-protein water-soluble molecular ion in the signaling pathway. Its small size and water solubility is important because it's going to allow it to spread rapidly by diffusion. So we have cyclic AMP, which is cyclic adenosine monophosphate. Adenylyl cyclase is an enzyme in the plasma membrane that's going to convert ATP to cyclic AMP in response to the cellular signaling. So here you can see this adenylyl cyclase is going to take these phosphates here, and it will go from being ATP to cyclic AMP. So you can see where there's 3 phosphates here to just 1 phosphate. And then phosphodiesterase would convert it to AMP. So in this example, you have your first messenger, which is your signaling molecule. An example would be epinephrine. It will come in and it will bind to this G protein coupled receptor. It's going to send the G protein over. It's going to activate the adenylyl cyclase, which will turn ATP into cyclic AMP, and that diffuses as the second messenger to go to this protein kinase A. An example of something that would do this is epinephrine. It will bind to the receptor protein, activates the adenylyl cyclase, and then you'll synthesize many cyclic AMPs to allow amplification. And then phosphodiesterase would convert it back to AMP to end the signal. So this is going to activate other proteins, usually a protein kinase A that phosphorylates proteins. So inhibitory G protein is going to inhibit the adenylyl cyclase. It gets activated by a different signaling molecule. So you want to remember that you have your protein kinases and protein phosphatases that go together, and your adenylyl cyclase and inhibitory G proteins that are going to work together. Another way we can do this is using calcium ions and inositol triphosphate. So these signals in animals will induce responses in targets using pathways that increase the concentration of calcium. Calcium can act as a second messenger since the concentration in the cytosol is normally lower than outside. So the calcium levels inside the cytosol are going to rise by a mechanism that releases it from the endoplasmic reticulum. And then the inositol triphosphate and diacylglycerol are going to be the messengers produced by cleaving them in certain phospholipid membranes. So this is just showing an example of one here. So just know that calcium can also be a way of acting as a second messenger. So the response, this is going to lead to regulation or cytoplasmic activities. So you can have nuclear responses and cytoplasmic responses. So you start out with reception, then you're going to transduce this a lot of times through a phosphorylation cascade or as a second messenger, and then you're going to have the response get carried out. So in this example here, it's going to activate a transcription factor that's going to end up turning on a gene. So these things may regulate the synthesis of a protein or the activity of a protein or other cellular attributes like shape. Some pathways will lead to cell division, so if these malfunction, it can lead to cancer. So why have so many steps in this? It allows for fine-tuning of the response. Multiple steps allow for places for amplification, it allows regulation points, and places to coordinate things. So with signal amplification, at each catalytic step, there's more activated products than there was at the preceding step. Also, the specificity of the cell signaling and coordinating the response is important here. So in this first example, you have a signal molecule that's going to come in, bind to the receptor. The relay molecules will pass the message down for a single response. In this example, you have the signal molecule comes in. While it's being passed down the chain, you can actually have it lead to different responses, multiple responses. You can have one pathway going on where you have the signal come in, it's going to pass it down the cascade, and a second signal will activate another protein, and that cascade can come in and act with the regulation here. Oh, it's all right. Thanks. So it will activate or inhibit this pathway. And then finally, you can have this same signal molecule. It's going to attach to a different receptor this time and lead to a different response. So different cells can have different proteins, so the same signal can trigger different responses in different types of cells. You see this in our bodies with epinephrine. It's going to trigger the fight or flight response. So in the liver, it's going to cause the cells to break down glycogen. In the heart, it's going to cause the cells to speed up and contract faster. And that's a more efficient way of regulating it if you have one signal that's going to tell each cell to do its own specialized job that it's supposed to do in that circumstance. So one of the things that gives us some of the signaling efficiency are these scaffolding proteins and signaling complexes. So if you think about what scaffolding does on a building, it holds the workers up in position so they can do their job. The scaffolding proteins are large relay proteins to which several other relay proteins can simultaneously be attached. This makes things more efficient, allows the enzymes to find the substrate instead of relying on diffusion because it puts everything right in the immediate proximity. So these scaffolding proteins can permanently hold together networks of signaling proteins at synapses in the brain. So terminating the signal, in order to keep the cell alert and responding, these signal pathways only last a short time. When you bind the signal molecules to the receptor, it needs to be reversible so that receptor can revert back to the inactive form when the concentration of the signaling protein decreases, so you can end the signal. So apoptosis is one signal that's very important. It's a pre-programmed cell death. It's going to integrate multiple cell signaling pathways. So during apoptosis, your cellular agents get chopped up. It's going to chop up the DNA, the organelles, all of your cytoplasmic components. The cell is going to shrink and become lobed. We call it blebbing. And then the parts get packaged up into vesicles that are going to be engulfed by scavenger cells and eventually recycled. Apoptosis is also important during embryonic development. It's critical for the regulation. So the caspases are the main proteases of apoptosis. They're going to get activated when the cell receives the death signal. So in humans and mammals, there are several pathways involving about 15 different caspases that can carry out apoptosis. Whichever pathway is used is going to depend on which type of cell it is and the trigger. So a lot of times the signal is going to originate outside the cell from something like a neighboring cell. The signal can also originate inside the cell. It can come from the nucleus when it looks and says we've got irreparable damage to the DNA. Or in the endoplasmic reticulum with excessive protein folding. It says, you know what, this is just too much for us to deal with. Let's scrap this cell. It's important for developing paws and feet. So lower levels of apoptosis are found in the web feet of ducks. In humans, you're going to have apoptosis when you look at an embryo or fetal development. You're going to start to see the fingers, but early on, the fingers appear webbed. Failure of apoptosis can result in webbed fingers and toes in humans. So there's evidence suggesting that this is also going to be involved in certain degenerative diseases like Parkinson's and Alzheimer's. Some forms of cancer can result from a failure of apoptosis. So at this point, you have probably already seen several things that can lead to cancer in the cells. Cancer is not just one disease, it is a series of diseases. So keep that in mind when you start thinking that there might be one thing that's a cure for cancer. Multiple things can go wrong to lead to cancer.